All right, so we're going to, for the next 45 minutes, give John opportunity to talk about why and how we save seeds, and then 15 minutes of questions. And I'll just give you the forewarning that after that we have 2 presentations that we are going to give time for, so we will keep it within that time.
All right, welcome everyone again this afternoon. So the topic that I was asked to speak about is why and how. But I believe, first of all, I am not qualified to talk about how. I do have some very limited experience, but if you want to get into the conversations about how, and the reason is the how to save seed is, first of all, it's much, much simpler than you might think. It is not that difficult. But there is a great deal of diversity across different species. If you're saving seeds from cucumbers versus tomatoes versus other crops, there are a lot of different species-specific information that you really need to know. It's not difficult, it's not complex, it's just species-specific. And so if you want to have the how conversation, you should have a conversation with my brother Emmanuel outside at Sower Seeds exhibits. And if you're really serious about getting into it, buy some of the books that he has that describe the how. And really, I believe it is most important to have the conversation about why. Why do we need to save our own seeds? And this is what I am going to focus on most. And I'm also happy to entertain any questions about how that I might be able to answer. So, why should we save seed? In Scripture, we read very clearly that the sins of the fathers will be visited on the children's generation until the 3rd or 4th generation. And that he will provide mercy to many people who love him, to those people who love him. And there is this science called epigenetics. And the science of epigenetics emerged from the realization that we don't actually know what we thought we knew following the Human Genome Project. So in the Human Genome Project, the belief, the expectation was that we would study DNA, and once we understood the DNA structure, the genetic structure of a human, or of any other organism for that matter, scientists believed that the cause of dis-ease, the cause of illness and disease, would be a genetic problem, and that if we could just simply fix the genetics, then we would solve all of these degenerative illnesses like diabetes and heart disease and stroke, cancer, etc. Well, we came to the conclusion of the Human Genome Project, and we realized that there wasn't nearly enough DNA structure present to describe all the variability that existed. And that the way we had been thinking about DNA as a code to code for information couldn't possibly work. Do you know that our DNA overlaps 70% with the DNA of a corn plant? 70. 70% of the DNA of a corn plant has segments that are exactly identical to our own DNA. Isn't that remarkable? So that is an illustration that there was not enough stored information as it was understood and perceived then on the DNA to describe genetic inheritance, to describe genetic heritability. And from that emerged the science that is called epigenetics. And the simple summary of epigenetics is simply that environment determines genetic expression. Environment determines genetic expression. So what this means is, and we all know this from a practical perspective, we know it very well. You can get seed out of the same bag and plant it into 2 completely different soil types, one that is an optimal soil type and optimal fertility, optimal growing conditions, and another that is much more challenged, and the way those plants express themselves is completely different. That is epigenetic expression in the parent generation. But then, what is really fascinating is how epigenetic expression expresses itself in the future generations coming from that parent generation. And I'm going to give you a few non-plant examples. About 18 months ago, a group of scientists released a study that they had conducted on mice to evaluate the long-term generational impacts of feeding tiny doses of glyphosate to the parent generation. So the first generation of mice was fed grain that contained 10 parts per million glyphosate, 10 parts per million of the active ingredient of Roundup. And to give you some frame of reference for 10 parts per million, the acceptable threshold for corn in North America is 400 parts per million. The acceptable threshold for soybeans is 600 parts per million. The acceptable threshold for garbanzo beans is 4,000 parts per million. In other words, levels that are significantly higher than this 10 part per million threshold that they were using in the test grain. So the only mice that were exposed to glyphosate in their diet was the first generation. The second, the third, and the fourth generation in this trial were fed grains that were measured in a laboratory and were known to have non-detectable levels of glyphosate. The first generation had some comparatively mild negative health impacts. And when I say comparatively mild, I'm talking about 30 to a 40% increase in the incidence of cancer. So there were some very significant health consequences for the parent generation. The second generation had less incidence of disease than the parent generation. But guess what happened to the third and fourth generation? The third and fourth generation had an incidence of cancer that was 3 times greater than the parent generation that got fed the glyphosate, even though their glyphosate in their diet was 0. That is epigenetic expression. That is visiting the sins of the fathers on the sons and daughters for the third and fourth generation. You know, when you are sitting beside a lady, you're sitting beside 5 generations of women. When you laugh, but it's true. When a baby girl is born, the ovaries for her future children have already been formed. So the diet that a mother is exposed to and the toxins that a mother is exposed to when she is pregnant have an impact on the ovaries of her unborn children, on her grandchildren. So in the case of these mice, the impacts on the third generation were significantly greater than they were on the second generation because the third generation was at the ovary stage when it was exposed to tiny traces of glyphosate. Now, I'm giving you this example because it's one of the better researched and studied examples. We don't usually, there aren't very many studies, in fact there are none that I'm aware of, that study the positive impacts of epigenetics in plants over several generations. Everyone is studying and looking at and evaluating toxins, and there's not many of those studies either. But this is a clear expression of how exposure to toxins can affect future generations. And societally, culturally, around the world, we are now, frequently when we have conversations about bad habits that people have, like smoking and drinking, etc. This is a particularly relevant conversation for smoking. One of the criticisms that comes back is that, well, my grandfather smoked and he lived to be 90. What's different? What is different today is that your grandfather and your grandmother were not exposed to the toxins in the formative years, in the first decade of their life, that children are being exposed to today, from in fact even before they were born. And it is because of that today's generations and future generations, we have life expectancy in the United States is on the decline for the last 5 years. And 2 years ago, the CDC reported a statistic that should have freaked people out, but it barely made the news. 2 years ago, the CDC said the children being born today are expected to die before their parents. Think about that. That is partially, not exclusively, but this is partially an agricultural problem. farming and agriculture. There's also food processing as well. Agriculture alone is not alone to blame. But this is partially a food and agricultural problem because we are spraying and coating our food with toxins. That is impacting the third and fourth generations and perhaps longer into the future in ways that we do not yet understand because we haven't been looking for the information. So, enough of that. Let's switch it to the positive. This same process that I've been describing for transferring negative impacts of toxins to future generations also holds true in reverse. That when you begin managing the parent generation for optimal health, for optimal nutritional support, for optimal microbiome support, that also transfers to the second and third and fourth generation. That is the hope. That is the inspiration. That is the opportunity that we have. So, I want to begin by describing agronomy and plant nutrition through the lens of biology instead of through the lens of chemistry. For the last 120 years, agronomy and plant nutrition has been measured and managed through the lens of chemistry. We talk about maintaining soil mineral balance and soil nutritional balance and measuring plant sap analysis and tissue analysis from a mineral balance perspective. And that chemistry conversation has defined the conversation for the last 120 years for the very good reason that it was what we could easily measure. We didn't have good tools for measuring the soil microbial profile. So we measured soil balance and plant balance in terms of chemistry. And if you read any plant nutrition handbook or any white paper describing certain nutrient interactions, there is always the obligatory introductory paragraph or introductory article describing how plants absorb nutrients. And the conversation goes something like this. Nutrients are held in the soil solution, in the soil water solution as soluble mineral ions. In other words, as soluble salts in the soil solution where we have magnesium ions and calcium ions and potassium ions. And that these water-soluble ions move through the soil solution, through the water solution, to the plant roots. They're absorbed by the plant roots and then transferred throughout the entire plant's vascular system and used as a nutrition source. Real-world agriculture has never worked that way, ever. This is a glorified hydroponic model. And if we rely on this system in an outdoor agriculture environment, we are depending on a glorified hydroponic system. It is true that plants have the capacity to do this. After all, this is how hydroponics works. But this is not how living systems actually work, and never has been. It's not the way they were designed to work from an optimal health perspective. If you have traveled, and if you have paid attention, many of us have gotten to observe trees and plants that are growing in what appear to be impossible conditions. I have observed in a number of locations all across North America trees growing out of hard rock where there is no soil. There is no soil and there are no water-soluble nutrients. So how do they get nutrition? That's the question that I have been asking for 20 years. And there have been hints in the research. Some of the preliminary research was done in the early 1900s, but it's really only been in the last decade that we now are developing deep insights into how biology-centric plant nutrition actually works. And it is such a beautiful process. One of the leading and more innovative researchers in this space is named Dr. James White. He's at Rutgers University in New Jersey. And he has led a group of colleagues from around the world to evaluate and to study this process that they call rhizophagy. Translated into plain and simple English, rhizophagy means root feeding. And I wish I had a marker board, so I'm going to try to build an image in your mind. Please follow along with me here. So we have what Dr. White and his colleagues have described is that as you have a growing root tip that is growing and extending through the soil profile, Imagine that you have this growing root tip that is as a pipeline moving through the soil profile, and the very tip of the root itself is porous, like a sponge. And we now know that bacteria and fungi and algae, at least those three species and possibly others, can move through the root tip directly into the plant root, and they do so in extremely large quantities, on the order of millions of bacterial cells per day, moving into every individual root tip. And then these, for the sake of this conversation for the moment, I'm going to use bacteria and focus specifically on bacterial populations, but everything that I'm describing also holds true for fungi and algae, at least those two that we know of. Then these bacterial cells, as they move back from the root system, or back from the growing root tip, the plant releases reactive oxygen species and nitrous oxide. It's a very oxidizing solution starting about a ½ inch back from the growing root tip. So think of it as being exposed to hydrogen peroxide. What happens in this very oxidizing environment? Why, thank you. Can you use that? Sure. So what happens is, in this oxidizing environment immediately behind the root tip has the effect of stripping off the cell membranes of the bacteria. And now we have this naked cell protoplasma. And now these naked bacterial cells that no longer have a cell membrane move back, they move throughout the plant's entire vascular tissue. They will go, there is no place on the plant where these bacteria do not go. They will go into the seed, into the fruit, into the blossom and pollen, inside the leaves, on the leaf surface. You know, in our bodies, thank you, the only 10% of the cells in our bodies contain human DNA. 90% of the cells in our body contain bacterial DNA. We are more bacteria than we are human. Can you imagine that? The same is true of plants. These bacterial populations colonize the entire plant profile. And then, you're expecting me to be a reasonably good artist here, which is a very lofty expectation. So this growing root tip is porous. These bacteria move back through this extending root tip, and then at a distance of roughly 2 inches behind the root tip, some of these bacterial cells survive this process of being exposed to this oxidizing environment, and they begin forming these small clusters of bacterial cells. And once these small clusters begin to form, this triggers the formation of what we call root hairs. And then something marvelous begins to happen. The plants, these small clusters of bacterial cells begin moving out through these root hairs back into the soil environment. And the plant releases not just these bacterial cells, but it also releases a mucilage and a mucigel that contains the exact compounds that these bacteria need to rapidly reform their cell membranes. So the plant is not just releasing the bacteria, but it's also releasing the exact food sources and the exact materials that they need to thrive and proliferate. Now, as these bacteria are making this journey from the root tip back to the point where these root hairs are being formed, the plant is also communicating to these bacteria, both through chemical signaling pathways and through electrical signaling pathways. And it is telling these bacteria, I need more phosphorus, I need more manganese, I need more zinc. It is very specific for each precise nutrient that this plant requires more of. And then these bacterial cells now go back into the soil environment, and in a healthy soil, there's a key caveat, in a healthy soil with an abundant microbial population, once a microbial population becomes large enough, this does not happen in unhealthy soils that do not have good biology, but once the population becomes large enough, the the whole population begins behaving as one organism instead of a multitude of organisms. This is important. This process is called quorum sensing. And what quorum sensing is, the same thing happens in causing infectious disease. So if we have I'm blanking out right now on the right word. If we have an infection that causes a biofilm is an expression of quorum sensing, because what happens is you have a large enough cluster of bacterial cells, let's call it, for the sake of discussion, 5 million bacterial cells. But all of a sudden, up to that point, there is a threshold, there's a very clear threshold that determines quorum sensing. So below 5,000 cells, each one of those bacteria behave as an individual. They do everything for themselves. They don't rely on anyone else to do anything else. But once they cross the quorum sensing threshold, now some of the bacteria on the outside of the cluster begin forming protective membranes and protective compounds to protect the cluster. That's where biofilms come from. The bacteria on the inside of the cluster do something else entirely. They do something different. So there is this division of duties that occurs once you have a large enough population to cause quorum sensing. But the reason this is important, the reason I'm describing quorum sensing, is because quorum sensing facilitates practically instantaneous communication throughout the entire soil microbial community. This happens so rapidly. There are some fascinating studies that were conducted on soils where they cultured bacteria in the lab that were they developed antibiotic resistance in them by exposing them to antibiotics. And so these bacteria now contain the genetic information of how to be resistant to antibiotics. When they add those bacteria to soil, that genetic information is exchanged, and the entire soil microbiome will contain the genes for genetic resistance to antibiotics in 48 hours.
How big an area?
4 feet deep.
And how wide?
4 feet in diameter. 48 hours. But in terms of quorum sensing, so that's actually, I sidetracked a little bit. That is slightly different. This is exchange of genetic information. That's slightly different from quorum sensing. Quorum sensing is instantaneous. So now you have these bacteria move out through the soil profile, and they have received the signal from the plant. Plant says, I need more zinc. They move out through the root system. They come in contact with the rest of the soil microbial population, and they send the signal, the plant that is feeding us sugars needs more zinc. Go get more zinc. And immediately, the entire population, for a radius of yards around that plant, those microbes all of a sudden begin extracting more zinc from the soil mineral profile and accumulating more zinc inside their cells and moving more zinc into the plant. Isn't that amazing? You know what this means? Plants are farming bacteria and organisms. They have a symbiotic relationship, and they're farming bacteria and organisms exactly the same way that we farm livestock. It also means plants are not vegetarians. This is a beautiful, beautiful process. It's a beautiful system. This is the way plant nutrition was designed to work. This is the way it works in natural ecosystems. When we have forests and pastures that are not fertilized, this is how plants absorb nutrients. This is how the trees growing on a bare rock surface are absorbing nutrients. It is through the action of biology. And I love this process. There's so much more that I could talk about here. But the bacteria and the algae, yes, they are able to get minerals out of the rock. See, there is this symbiotic relationship between plants and the soil microbial community. The plant is supplying sugars. When you think about sugars, I'm going to expand on this point for just a bit because it's important to understand. When the plant releases sugars to feed the soil microbial population, what is sugar? What minerals are contained in sugar?
Carbon, hydrogen, and oxygen.
Yes, exactly, and nothing else. Carbon, hydrogen, and oxygen. There is no zinc, no manganese, no copper, no calcium. There are no other minerals in sugar. And yet, the bacterial population needs minerals to form their own cells. So what do? They extract, they etch those minerals out of bare rock and incorporate them into their own cells. And then those bacterial cells are transported back to the plant and provide the plant with nutrients. And by the way, this process that I'm talking about, I haven't, for simplicity's sake, I have not included mycorrhizal fungi and others. But now, if you want to include mycorrhizal fungi in this, imagine that you have this mycorrhizal fungi which is tapped into this root as a pipeline. And this pipeline contains millions of bacterial cells. The beauty of mycorrhizal fungi is that you have this really large hyphal network that is significantly larger than the plant root system itself, and it is, I was about to say it's nothing more, it is more than that, but one of the major features of this mycorrhizal fungi network is that it is a pipeline for transferring bacteria from the soil population directly into the plant roots. So let's say you have soil where you've determined that your soil is deficient in zinc, and you make a zinc sulfate application, and the amount of zinc sulfate that's required to solve your soil's nutritional imbalances is 5 pounds per acre, and you have two options. You can apply zinc sulfate in dry pellets, where if you spread 5 pounds over an entire acre, you will end up with 1 pellet for every 3 square feet, or every square yard. Or the other option is that you can dissolve zinc sulfate in water and spray it on so you get much more uniform distribution. The second option is much more challenging for many growers. It's much more easier for most growers to do a dry broadcast application. It is problematic because you have a distribution problem if you're relying on a chemistry-based system. But when you have this fungal hyphae network, they can extract zinc that is 20 feet away from the plant and make it available to the plant very efficiently and very effectively. Anyway, why does this matter? And why did I speak about this in an epigenetic context? Because this rhizophagy cycle is broken in most of our modern genetics and varieties. Severely dysfunctional. When we think about propagating seeds for future generations, actually, I'm going to give you an example. An individual named Walter Goldstein at the Mondawmin Institute in Wisconsin has been developing and breeding corn varieties for the last, I'm not sure how long Walter has been at this, at least a decade, if not long, actually probably closer to 2 decades that I'm aware of. Walter found some teosinte and some old corn genetics in Mexico, Central America, that contained bacteria inside the plant that had the ability to fix nitrogen and provide all of the nitrogen to a corn crop with no further additions of nitrogen required. And he has been developing these corn cultivars, developing different varieties and breeds, and growing them out in challenging conditions, and this I'll speak more about this in just a minute from a breeding perspective. But he has identified that, in collaboration with Dr. White, that these corn varieties that he has developed contain a number of different bacterial populations that are capable of fixing nitrogen without the need for legumes. And he's also identified that those particular microorganisms, those particular bacteria, can be transferred from his varieties to other corn varieties. They can be moved from one population to the next. It is my understanding, I'm not aware that this research has yet been done, but it's my understanding that if you want to prevent those bacteria from functioning well, if you want to shut them down in one generation, then it's very easy to do so. You plant the seed and you add nitrogen fertilizer, and that bacterial population will die. Why? Because The plant and the microorganisms have this symbiotic relationship. And a symbiosis means that it is a win-win. Both are contributing to the relationship and both are receiving value from the relationship. And in the case of the symbiosis between the corn plant and these bacteria, the corn plant is providing sugars and the bacteria is providing nitrogen. There is an equal exchange. The moment the corn plant can readily absorb excess of soluble nitrogen from the soil profile, it tells the bacteria, hey, I don't need you anymore. Why would I waste sugar feeding you? I have all the nitrogen I need. And that bacterial population dies. Over the last 60 years or longer, we have completely ignored biology in our breeding work. And as a result, many of the modern genetics and the modern varieties that are available to purchase today no longer have the optimal association with these microorganisms that can provide this rhizophagy cycle, that can provide these crops with nutrition in the absence of fertilizer. If I were to say it differently, I would say that our modern genetics, the seeds that are available today, have been bred to require the use of fertilizer. They are dependent on it. They are drug addicts. Genetically, drug addicts over multiple generations. Remember my conversation about epigenetic expression over multiple generations? They now have multiple generations where they are completely dependent on chemistry fertilizer. And if we try to remove the chemistry fertilizer from the system, those particular varieties crash and burn. They are not adapted to thrive in a biologically active, healthy soil. To come back to Walter Goldstein and his breeding efforts on corn, in an effort to preserve and to enhance the ability of these nitrogen-fixing bacteria, all of his corn selection and breeding work is being done on soils that has no applied nitrogen and has had no applied nitrogen since the beginning of his research for the last 20 some odd years, and also has no applications of manure, because manure can also provide excess of soluble nitrogen. So I'd like to provide you with another story, another example. Roughly 15 years ago, when actually it's a bit longer than that, but roughly 20 years ago when I started doing consulting work, one of the organizations that I collaborated with a little bit was an organization called Midwestern BioAg. Some of you may have heard of Gary Zimmer. And a few years into working with them, they developed a much closer relationship and collaboration with an alfalfa breeding company called Cal West. And the conversation that Midwestern BioAg had with Cal West was to say, look, you are breeding and selecting alfalfa varieties to thrive in a system where there is the constant application of potassium chloride fertilizers, and the soil is very hard and compacted. There are herbicide applications. That is not our system. We want to work with you to select alfalfa varieties that are optimized for the way that we manage these biological systems. Soils that are biologically active, where manure is being applied, where limestone is being applied, compost is being applied, cover crops are being grown. And we want to work with you to select alfalfa varieties that are adapted to that system. And so they did. In 3 years, must rank up there amongst the most successful, rapid success breeding programs out there ever. In 3 years, they released several varieties from this new selection program where these varieties were adapted to thrive particularly well in these biologically active soils. One of those varieties was called Winter King. Winter King alfalfa is now at least on its third and maybe on its fourth generation. I think you can now today buy Winter King 3, possibly Winter King 4, I'm uncertain. Winter King alfalfa was so productive and so successful. It outperformed all the other alfalfas in a biological system by a very wide margin. But not only did it outperform in a biological system, it also beat every competitor in multiple trials across multiple regions in the mainstream system. because of epigenetic expression. Because here was a plant that was selected and adapted to have a close symbiotic relationship with lots of different microorganisms that, and here's the key piece that I haven't gotten to yet in this conversation, these microorganisms are intended to be vectored on the seed. They are intended to be carried on the seed from the parent generation to the subsequent generation. And this is one of the ways that the loss of these microbial symbionts with plants has been lost, is that today, for many different species, not all, but for many species, cell culturing is often a part of the propagation process. And in cell culture, we put plant cells into a petri dish in the lab, and we soak it with antibiotics to kill everything except those particular plant cells. So we kill all the bacteria and everything that is associated with that plant as a part of the propagation process. And this is why modern corn genetics have almost universally, at some point in their propagation cycle, were propagated using cell culture. So modern corn genetics contain none of the bacterial populations that are contained in Walter Goldstein's corn. None of them. This means, so when we have our modern genetics that do not have these microbial associations in the seed being carried on the skin of the seed, it means when those seeds are planted into the soil, in order for this process to work, they first have to recruit bacteria from the bulk soil environment. That is a process that takes time. It takes 10 to 14 days usually after a seed germinates before it can effectively recruit bacteria from the soil profile, because it first has to germinate, then it has to emerge, then it has to begin photosynthesizing and producing sugars beyond the point that is required to sustain itself. And only at that point, usually 10 to 14 days after germination, it can start sending sugars out through the root system as root exudates. And it's only at that point, once it can start feeding bacteria, that it can recruit bacteria. And that initial 2-week window is when most diseases infect a plant for the first time, because there is no protective coating. There is no protection in the rhizosphere. So we live in a world today where many people are beginning to recognize and realize and appreciate the importance of having biologically healthy soils. We have a proliferation of information on how to produce healthy, high-quality compost, and how to produce compost teas, and worm teas, and worm extracts as biological inoculants to add to our seed and to be used when we are planting. We now can buy mycorrhizal fungi inoculants, and we can buy microbial inoculants such as Rhizobium bacterium, bacterial inoculants, and so forth. But there is this little secret that most people in the biological community do not talk about because they don't have an answer for it. And the secret is that 95%, greater than 95%, of all of the beneficial bacteria that are required for a plant to have an optimal functioning immune system and be resistant to soil-borne diseases cannot be propagated in a lab. And they cannot be propagated in a compost pile. And they cannot be propagated in any known method, with one exception. They must be propagated in the presence of a living plant root. They must be propagated in the presence of a living plant root. So what I'm describing is that we should think about, when I think about microbial inoculants that we can use to rapidly regenerate soil health, we use a lot of microbial inoculants in our work, and I'm a big fan of them. They are very effective. We use compost teas, vermicompost teas. We use purchased microbial inoculants. They are not a replacement for the bacteria that is supposed to be vectored and carried on the seed from the parent generation to the following generation. There is no substitute for that. You cannot buy a substitute for that. You have to grow it in the seed. So let's talk a little bit about seed production. What time was my supposed to end? 5? cats. Let's talk a little bit about seed production. So my experience is not so much with small-scale market garden, home gardener seed production. I certainly know many people who save seeds. I grew up, my parents saved seeds. But specifically, I'm speaking specifically to large-scale commercial seed production of vegetable seeds, of corn seeds, and so forth. Seed production is considered to be something you do on your worst soil that you cannot grow an economically profitable crop with otherwise. I still remember being in a conference in Indiana, probably 15 years ago at this point, sitting at large circular tables eating lunch. There's a group of probably 8 other people at the table, and there's a conversation on the other side of the table. And this farmer makes the comment, he said, yeah, well, this, this one field, it doesn't produce enough yield to justify putting it into a regular corn crop, so I just use that as my seed corn production field. And I went, what? What did you just say? When we think about epigenetic expression and the sins of the fathers being visited on the 3rd and the 4th generation, we should seek to grow seed that is the largest, heaviest, most nutrient-dense seed that contains the greatest microbial population possible. In other words, we want to grow seed in an absolutely ideal environment, because the nutrition contained within that seed and the microbial population contained within that seed, when then planted into more difficult environments, allows it to thrive more effectively, as in the example of Winter King alfalfa. Winter King alfalfa was developed, or selected, I should say, in an optimal environment from a microbial health and mineral balance perspective. And that produced such a pronounced epigenetic effect that the results lasted for multiple future generations. This is what we need to seek to replicate in our own seed-saving efforts. We need to provide our parent seed, our parent crop, with optimal nutrition and with an optimal microbial environment. And when we think about selecting seed, this is something else. I'm on a bit of a soapbox here, so I'll just keep going. The way commercial seed corn is produced, seed is one case where bigger is almost universally better. Bigger seed, heavier seed, is seed that contains more nutrition, more sugars, more carbohydrates, more energy. It is a healthier seed that will germinate faster, that will emerge faster, is more stress resilient, and can vector and carry more bacterial populations with it to the next generation. So when it comes to seed size, we want to select the largest, heaviest, highest test weight seed we can possibly find. Except in seed corn, they do the exact opposite. Seed corn now gets sold by seed count. And there are logistical efficiencies for the farmer and when planting in the spring, and for the seed company to have smaller sized seed. So the process today is, first we plant seed corn on soil that has poor nutrient content. It's not fertilized very well. No microbial inoculants are added. Then, when it approaches the silking stage or the tasseling stage, they now use mowing machines as detasselers. They move through and they detassel the corn plant immediately above the ear. And they remove the top 1/3 of the plant. So they limit the plant's photosynthetic ability to produce sugars. They cut the plant's ability to produce sugars by 1/3. So now you have fewer sugars going into the kernel. Then they allow the seed to mature. They do not want it to approach the black layer stage, or the stage of physiological maturity. So the moment the seed is considered to be large enough and starts just approaching maturity, they will desiccate the plant with an application of sodium chlorite, not chloride, but sodium hypochlorite, which is a very powerful toxicant. They will desiccate the plant, and these desiccating agents that they use they will penetrate through the husk on the ear. They will completely coat the seeds, and they are a very effective microbial sterilizing agent. So you end up, by design, not by accident, but by design, they end up with a plant that is a seed that is deliberately small. They want small seeds, not large seeds. Small seeds that are compacted and shriveled up, and there are more seeds in a bag. A 100,000-pound seed count bag used to weigh 60 to 70 pounds, and now it weighs 30. And I'm using corn as a very well-known example, but what I've just been describing is true for many different crops, modern crops, and the way that they are selected. So the reason why we need to save our seed is, and this is not about the how, as you can tell, but the reason why we need to save seed is to not just to preserve what has been lost, but to regenerate some of those things that may have never existed, or that existed only generations ago. We need to save seed because when we look at epigenetic restoration, so think about the Winter King 2 alfalfa example. There's a few other examples that I could go into as well, but time doesn't permit that. But when you think about the rapid improvements, there are many people. Actually, I'll give you one example. A grower that I worked with in Massachusetts a number of years ago got the same variety of arugula from 2 seed sources. One seed source didn't have enough to fulfill his order, so he filled in from a second seed source. It was the exact same variety, grown in the same growing year, from growers that were about 20 miles apart. So the seed production was about 20, same general region. You would expect similar climate, similar weather impact stress, but very different nutrition management. The seed from the one grower was almost double the size. They only had half the seed count per pound as the seed from the second grower. And when they planted these arugula seeds at the exact same time, same management conditions, the larger seed produced arugula that was 4 times larger 10 days earlier than the small seed. That is a remarkable expression. So this is not breeding. What I just talked about, the effect that I just described is not a result of multi-generational breeding. It was simply nutrition, epigenetic expression as a result of nutrition management on the parent generation. Now think about what that could have looked like if they then saved seed from those large, vigorous plants. And they repeated that over multiple generations. Now you have epigenetic compounding where it's getting better and better and better instead of getting worse and worse. Yes, Roman?
Can you speak a little bit to the advantage of planting seed in your own soil where you're going to grow and the epigenetic value that brings?
I'm not sure where you're going or what you're referring to.
Okay, well, I understood that when you let a pasture go to seed, the seed that it drops is better than what you planted to begin with.
Yes.
And
Why?
it keeps getting better.
Yes. Why?
That's what I want you to explain.
I was having this conversation a week ago with a group of cover crop. Actually, no, it was the beginning of this week with a group of cover crop seed producers in Nebraska and Colorado, and with Keith Burns from Green Cover Seed, and he was describing how rapidly seeds and crops can adapt to a local environment and to local climatic conditions and to your specific soil. And the example that he used is they had a specific variety of cereal rye called Elbin rye that a farmer in Colorado really desired and appreciated because it began greening up and growing very early in the spring. And that was a trait that he specifically needed for his crop management system. And so he purchased Elbin rye and was using this purchased rye seed on his farm. And then they suggested that he start growing his own cover crop seed and just harvesting it himself, which he did. And in 4 generations, there was already a significant loss in the 3rd generation. By the 4th generation, it had completely lost that early green up. It had adapted to the Colorado environment. So this was a variety that was being grown in Oklahoma and was transferred from Oklahoma to Colorado. And in 4 generations, it had completely lost the characteristic that he was specifically looking for because it had adapted to the local climate. It had adapted to the local ecosystem in 4 generations. That's how rapidly that plants can adapt to local ecosystems. And so what I'm referring to, what I'm describing there is climactic adaptation, which is very real. But what Roman is also referring to is microbiome adaptation, where if you grow seed in a certain soil and the microbiome becomes associated with that plant, what then happens is that seed gets, when that seed is planted in the same soil that it was grown in, or that the parent generation was grown in, there is, I'm trying to think of the best way to describe this simply. I described quorum sensing a little bit in terms of the microbial population. When a seed is planted into an environment where the microbiome of the soil matches the microbiome that on the seed, it immediately recognizes, I am home. I am home. And it starts growing because it recognizes that I am protected. My memories, the memories that this seed contains from its parent generation, it doesn't have to discard those memories. It is continued experience, not new experience, if that makes any sense. Now, the point that I want to make, the point that I'd like to leave you with, is how easy this is, how rapid and how overwhelmingly positive the changes are. With this rye propagation, it was 4 generations. When you think about the length of time that breeders who do not work with God's design take to develop a new variety, if they started working with plants, listening to plants, working with God's design, instead of trying to do something that is contrary to God's design, think about how much more rapidly they would be able to achieve those outcomes. Winter King alfalfa took 3 years of selection. That is unheard of in the breeding world. Usually selections take decades or longer. It took 3 years. This is why when we work with God's design, this is not difficult. This is easy. And I would suggest that it is our responsibility as stewards of His creation to engage in this way. This is not a question of preservation, of preserving what is lost. That is an element of this. But it is more than that. It is also optimizing for the environment today. Because the reality is that the varieties that thrived 80 years ago or 100 years ago, we have a different climate today than we did 80 or 100 years ago. We need varieties that are attuned to the climate today, that have the genetic adaptation and flexibility and resilience for where we are today, not where we were 100 years ago. I think we really lose sight of the work that we should be doing as stewards if we emphasize exclusively on preservation. This is not about freezing genetics in a moment in time. It's about consciously engaging as a hyperkeystone species, as stewards, to evolve to a better future. All right.
So that would be an advantage to grow garden seeds in the 2nd and 3rd generation.
Yes, when you grow garden seeds, the 2nd generation, the 3rd generation, the 4th generation, you will have these compounding effects where a variety that you begin with, let's say it's beefsteak tomatoes. You start growing beefsteak tomatoes, you select the seed, save the seed, and you plant that same seed for the 2nd and the 3rd and the 4th generation. Your beefsteak tomato, assuming that you are selecting seeds consciously, deliberately, and with care, will be significantly better in flavor, in quality, disease resistance, and local climate adaptation in several generations than what you originally started with. It happens fast. It happens fast. Yes?
Can plants get water from the atmosphere?
Yes, plants can get water from the atmosphere, some easier than others. There's some interesting research the USDA conducted back in the 70s. Corn plants, for example, are very efficient at getting water from the atmosphere. Alfalfa is not. There's wide species variation. But a corn plant can absorb water from the atmosphere all the way down to 15% humidity. So as long as the humidity is above 15%, a corn plant can get water from the atmosphere. Yep. Yes?
What about heirloom seeds? Are they under the same condition of not carrying the healthy bacteria that have been lost?
Very good question. The question was, what about heirloom seeds? Are they under the same condition of not carrying the bacteria that have been lost? And the answer is probably to a slight degree, but to a much lesser degree than the modern genetics. And maybe a better way of saying that is, I think it's a reasonable assumption, and we should operate under the assumption that many of them have lost some of that, because the genetic inheritance, the genetic memory, or maybe I should say the microbial inheritance of these seeds can be destroyed in a single generation when you fertilize plants with chemistry fertilizers. So if you're growing beefsteak tomatoes that are from 1850, and these seeds have been propagated, they carry these microbial associations, and you put on an excess of manure application, which 90% of gardens have, or you put on salt fertilizer, you disrupt some of that. Not all of it, but some of it. Maybe you lose 10%, maybe you lose 5%. And then that goes to another garden somewhere else. They make some other unknowing and unintentional mistake, and now you lose another few percentage points. And so over time, you lose it slowly and incrementally unless you plant it into extremely diverse, microbially rich soil, and then you can regain the majority of what was lost. Nature is incredibly resilient, and you can regain a lot of what has been lost in the past.
Do you have an example of seed saving where bigger was not better?
I can't think of one off the top of my head, no.
What do we do with animal manures then? What should be done with it?
Don't apply them. Do not apply them to your garden 6 inches deep. Animal manures are not inherently bad, but they are over-applied in excessive concentrations in gardens. That's the problem. It is excessive to the point where the nutrients become excessive, and excessive nitrogen or phosphorus or potassium from manure is no different to the soil microbiome than excessive nitrogen or phosphorus or potassium from a bag of fertilizer. It has the same chemistry effect and produces the same imbalances. Manure is not universally good. It can be applied to excess, as can anything.
Is it better if you compost it?
It is better if you compost it, but it can still be applied to excess because the nutrients are now less soluble, but they're also more concentrated. So if you put on 1 inch thick compost versus 6 inches thick manure, you've delayed the effect, but you've actually applied the equivalent of 12 inches of manure. So it's more concentrated but less soluble, and the effects aren't necessarily better. You can still overdo manure or overdo compost. Yeah. Yes.
So you spoke about the downside to modern seeds. What about seeds in the latent seed bank, and maybe specifically about quorum sensing to get them to come back?
Can you ask your question again in a different way? I didn't quite follow.
We were speaking some about quorum sensing, and I've heard, I don't know much about quorum sensing, but specifically using quorum sensing to revive seeds in the latent seed bank, such as
So you're asking, if I understand your question correctly, you're asking if you have seeds that are old, let's say they have been in storage for a decade or longer, and you want to regrow them and revitalize them, if they have difficulty germinating, can you improve their germination performance by putting them into a microbially healthy soil? Is that your question?
maybe more the seeds that have been in the soil for a long time that haven't germinated because of poor microbial soil.
Uh-huh.
How to get them to germinate again.
Uh-huh. You ask the most perplexing, fun questions. So the question, let me see if I can capture it accurately. The question is, when you improve the soil microbial population beyond the threshold where you now have good quorum sensing, will that stimulate the germination of seeds that have been dormant in the seed bank for a very long period of time? Is that your question? The short answer is yes, with profound and surprising effects. We are in the very early stages of testing a plant extract product, and I'm not at liberty to divulge the details because it's not our information to share. But they're extracts from a couple of different plants in a fairly concentrated form that were available as a byproduct. And they experimented with just spraying some of this byproduct solution onto flower beds, and all of a sudden there are what's the one that I'm looking for? Begonias. There are begonias and petunias and geraniums germinating all over this soil that the seeds were never planted. And in the case of begonias, there is no memory of ever having planted begonias. and yet the seeds are coming up from the entire soil profile. That was pretty remarkable, and that was an expression. I say this was applied plant extract, but we were able to measure and are in the process of further defining that this plant extract caused the proliferation of the soil microbial community. We built a very large microbial community very quickly, and all of a sudden, seeds started germinating that had been lying dormant in the soil for a very long period of time. And I'll end with one last thought. I asked a group of farmers the question last week: when a seed is planted into the soil, how rapidly, how quickly does that seed begin communicating with the soil and make decisions about germination based on what's going on in the soil profile? How quickly does that happen? And I ask the question because there is this idea that is communicated that a seed should be a self-contained unit, that a seed should contain enough nutrients that we can plant it into a challenged sterile soil, and it contains enough sugar energy and enough nutrients to germinate, to begin growing, and to begin photosynthesizing, all by itself with no nutrient input from the soil. And so the popular paradigm is that, again, this is the paradigm of observing or considering soil just a sterile substrate to hold root systems. The popular paradigm is that seeds don't need to communicate with the soil, and therefore they don't communicate with the soil. But we're forgetting this one phenomena that is known as the allelopathic effect. The allelopathic effect is that when there are some plants which transmit certain compounds through their root systems, cereal rye is one that is very well known, that when you plant seeds into soil that has a standing population of bacterial rye, and this particular seed or this particular crop is susceptible to the allelopathic effect, those seeds will never germinate. So it means the communication between the seed and the soil environment happens before a decision is made to germinate. I believe, from what I can tell, it happens in seconds. It happens very rapidly. That seed begins communicating with the soil microbiome. So I hope I've inspired you. I would encourage each one of you to go home. Don't overcomplicate it. Don't begin with more than you can achieve. Seed saving is not difficult, and I would encourage each of you to seek the experience. We need, as a collectively, as a culture, as a society, we need not just for a few individuals to hold deep knowledge, we need for many individuals to hold functional knowledge. All of us need to get experience in saving seed, even if we just save seed from one crop and propagate it in our gardens over multiple generations. It's something we need to do. The experience is invaluable. Thank you.